We're going to start this video by asking a very interesting question. So if our bodies have 61 different amino acid coding codons, how many tRNAs do you expect our bodies have? So normally you would probably think that because tRNAs have anticodons and anticodons are complementary to the codons on the mRNA, there has to be 61 different tRNAs, right? But that's not entirely true because we have 45 different tRNAs in our bodies, and therefore the only logical thinking we have out of this is that the anticodons of these 45 different tRNAs must accept more than one codon. And how is this possible? And this is explained by the Wobble Base Hypothesis. The Wobble Base Hypothesis really just states that, for example, if we have a tRNA with the anticodon U, C, and G, you would expect that the complementary bases on the mRNA will be adenine, guanine, and cytosine. But this is true as well, but there's another possibility. The other possibility is that the last codon can bind to another nucleotide, and because of this, these different tRNAs will be allowed to bond to different mRNAs or different codons in the mRNAs. And this is very interesting because this allows the different, like we said, we have 61 coding codons, and this allows all the coding codons to have a tRNA. And we also discussed how we talked about the amino acid being specific to the tRNA, or the tRNA is specific to the amino acid, excuse me, and then we can have more than one tRNAs for an amino acid. So that's very interesting, and let's understand the chemistry behind why this is possible. So what essentially happens is, the chemistry behind it is that the first two bases are supposed to have the Watson-Crick complementary base, but the last one can have an exception, and this is the Wobble Base Hypothesis. So let's quickly talk about the consistency with the overall theory of the tRNA. So this is still consistent with the fact that the amino acid, the tRNA is complement, or the tRNA is specific to amino acid, and this still aligns with our theories, or the theory we discussed in the last video. So let's quickly talk about another base called the inosine base. The inosine base is a modified adenosine nucleosome, where we have the amine. It's just an amine removed, and we will talk about this in a future video. But the only point of mentioning this right now is that you can just memorize this. The inosine is just a modified adenosine. Just memorize that. And we will quickly talk about the different possibilities in the Wobble base, because this can show up on exam, or it can be part of understanding a question. So without further ado, let's look at the entire possibilities. If we have C and A on the anticodon, don't be worried, because the only complementary base it can have is its normal complementary base, like we would expect it to. So cytosine will always have guanine, and adenosine will always have uracil. So here's how I remember this. This is probably not the best way to memorize it, but I just see it as cat. So the cat is always going to be the same. It's the same cat. So the T is an extra letter, but the CA just shows that whatever, if you see CA on the anticodon, don't be stressed. It's the same thing. But if you see anything else, that it can have different possibilities. If you see a guanine, you can either have a cytosine or a uracil. And if you see a uracil, you can have an adenine or a guanine. And if you see an inosine base, it can have almost everything, and that's including adenine, uracil, and cytosine. And these are the most common Wobble Base bases. So these are all thermodynamically stable. That's very important to mention, and that's why it's possible to. That's why we see this in the process of translation. So hopefully you understand what the Wobble Base Hypothesis is, and now you understand why we have 45 different tRNAs compared when we have more coding codons. So hopefully that makes sense, and stay tuned for the next video.